Hey guys, in this video we're going to be talking about transport, but before we get there we're going to just do a little review about the plasma membrane, the cell membrane. So if you remember, we have in our plasma membrane, we have all of these little phospholipids. This little guy right here, that's a phospholipid. Remember, the way that we've been drawing it is, let me scroll down just a little bit. We've been drawing it like this. Here we have the polar head. Remember, that's made of phosphate. That's going to be what gives it charge. We also have nonpolar fatty acid tails. And remember, we have our phospholipid bilayer. So we've got two layers of these phospholipids, and the polar heads are going to be oriented towards the outside of the cell and the inside of the cell, because that's where the environment is aqueous. And so polar things are going to be next to other polar things like water. Okay. The nonpolar fatty acid tails are going to always face one another. So they're going to be on the inside. So inside here of that phospholipid bilayer, facing one another because they are nonpolar. So they'll be repelled by the water and pushed together. And so that's actually going to be one of the things that helps to hold the plasma membrane all together. If you remember, we have different components in the plasma membrane. We have proteins. So this is a protein, this is a protein, this is a protein. This little squiggly looking one, that's a protein too. And remember, these proteins, they can have different jobs. Proteins can either act as receptors, sensing what's happening on the outside of the cell and telling the cell how to respond. They can be involved in support for the cell. You can see here in our picture that we've got, you can see here these little protein fibers. This is part of the cytoskeleton that's attaching to some of the protein. Or the proteins can be involved in transport, and we'll talk about that role today. We also have cholesterol. Remember, cholesterol here in the phospholipid bilayer. Remember, cholesterol is found next to those fatty acid tails, and so we deduce that cholesterol must be nonpolar because it's found next to those fatty acid tails. Remember, cholesterol helps to keep the membrane fluid. And we said that if cholesterol went missing, then the membrane wouldn't be fluid. If the membrane's not fluid, things can't pass through. If things can't pass through, the cell can't maintain homeostasis. And without homeostasis, the cell is going to die. Remember? Okay. And then our final component that we have here in the cell membrane, we have the carbohydrates. And remember, the carbohydrates, they are called either glycoproteins if they are attached to a protein, and they're called glycolipids if they are attached to lipids. But their role, they act as a name tag for the cell. They identify the cell, and it will let other cells in the body know what type of cell it is. So it's going to be it's a way for cells to be recognized for being a certain cell type. Okay, so that's our review of the plasma membrane. So we're going to be talking about transport today in the cells. And when we are talking about transport, we have really two categories of transport. Transport can be either passive, or transport can be active. Passive transport does not require energy. So passive transport, no energy is needed. Active transport requires energy. So today we're going to be focusing on passive transport, and there are three types of passive transport. We have diffusion, We have facilitated diffusion. And we have osmosis. We're going to talk about each of these in detail. For active transport requires energy. We have different types of active transport. We've got 3 different types that we're going to end up discussing, but that's going to be a topic for another class period. Today we're focusing just on the passive transport, the transport that does not require energy. So if we scroll down to look at my picture that I have here, the first type of transport that we are going to talk about is diffusion. And actually, let me move over just a little bit. So if you remember, when we talked about the plasma membrane and we talked about the nature of the plasma membrane, we said that the interior of the phospholipid bilayer is made of nonpolar fatty acid tails. And we said that small nonpolar molecules could pass through. I really shouldn't have a line there. Small, should be a comma, polar. Sorry. Small nonpolar molecules can pass through. And so we gave some examples. Oxygen is a small nonpolar molecule. Carbon dioxide is a small nonpolar molecule that can pass through. So when we're talking about diffusion, we are looking at molecules moving from an area of high concentration to an area of low concentration. So in this picture, we're looking at diffusion in a cell. And these little molecules would represent, again, something like oxygen or something like carbon dioxide, something that is small and nonpolar, and it can easily pass through the cell membrane. If you see in our first picture here, we have a high concentration of molecules on this side of the cell membrane, and there's a lower concentration of molecules inside the cell. So they are going to diffuse. We're going to have a net diffusion into the cell. Now, if you look, you can see we've got some traveling this way, the opposite direction. These molecules, simply due to kinetic energy, are going to travel back and forth through the plasma membrane, through that cell membrane, but we're going to have a net flow in the direction from the high concentration to the low concentration. On the other side over here, you can see in the cell, you can see inside the cell, there's more molecules on the inside of the cell, so like oxygen or carbon dioxide, and so they're going to diffuse outside the cell. Again, we might have some coming back in simply due to the kinetic energy of the molecules. They keep on moving, but they're going to spread out into the available space that they have. Okay. So what we say when we're talking about diffusion, we say that molecules are moving from an area of high concentration to an area of low concentration, or the phrase that we use is we say that it moves down the concentration gradient. Down the concentration gradient. That just means that it is moving from an area of high concentration to low concentration. And they will continue to move until the molecules reach a state of dynamic equilibrium. And so that means that the molecules are all spread out, but they keep moving. So the molecules are all spread out, but they keep moving. So dynamic means it's continuing to move. Equilibrium means that it's an equal concentration on in all areas. So in our example, we were looking at a cell. But diffusion can actually happen without a cell. Like if we were to spray like perfume in the middle of the room, all of those perfume, those molecules that make up the scent for the perfume, they would diffuse throughout the room. It would spread from an area of high concentration in the middle where we sprayed it to the corners of the room where there was a low concentration. And so that scent would diffuse. Or if we were to put some drops of food coloring into water, we could watch the food coloring spread out, and we wouldn't have to stir it or anything. The food coloring would spread out, and eventually the water would all turn the same color. So diffusion happens, it can happen in a cell, and it can happen without a cell. Our next type of diffusion, or the next type of passive transport that I want to talk about, is going to be facilitated diffusion. So facilitated diffusion, we're going to think about it as diffusion with help. Diffusion with help. So what types of things might need help diffusing from an area of high concentration to low concentration? It's going to be those things that are polar and charged, maybe like a sugar molecule, maybe like some type of ion. And because these things have charge, they're not going to be able to pass through the nonpolar fatty acid tails. So to get through the plasma membrane, they're going to have help. And help in this case comes in the form of a protein. So we've got two types of proteins. We've got, in this picture here on the left, this is called a channel protein. The one on the right, this is called a carrier protein. And the channel proteins, they're going to provide an environment that's going to allow charged molecules to be able to pass through. So the outside of the protein would have to be nonpolar. If you guys remember from our Chemistry of Life unit, when we were looking at amino acids, we learned that the R groups on those amino acids have different properties. So in this protein, the R groups that would be next to the fatty acid tails would be nonpolar so that they could be embedded in the membrane. But the inside of this protein, those R groups would have a charge. They would be polar to allow polar things like sugar and ions to be able to pass through the cell from an area of high concentration to low concentration. So this is still a form of passive transport. It does not require energy, and we still have things moving from an area of high concentration to low concentration. So if you look in our first picture, our channel protein, it's like a little tunnel that goes through the plasma membrane. You can see the little boxes. Let's imagine that that is our charged molecule that we're trying to get through. And you can see that they are moving from an area of high concentration on this side to an area of low concentration on the other side. Okay. The little star type molecules, they're not able to pass through because this channel protein is specific for this particular square shaped molecule. Over here on the right hand side, you can see we've got another protein. It just works a little bit differently. This one's called a carrier protein, and it literally changes shape as the molecules are passing through it. So if we look here, we can see we've got our green charged molecule. Let's imagine this is like a glucose maybe, and it's coming in. And once it is inside this protein changes shape and it pushes it out to the other side. We're still moving the little circles here from an area of high concentration to an area of low concentration. We're just doing it with the help of a carrier protein, which changes shape as things are passing through. So it changes shape after it interacts with that particular, the particular molecule that is passing through it. Next up, we have a special type of diffusion known as osmosis. And osmosis is going to be the diffusion of water. So osmosis is the diffusion of water. Water is going to always move towards the area where there are more solutes. Water moves to the area with more solutes. So solutes are going to be things that dissolve in water. It could be things like sodium chloride, like salt, or it could be like glucose or sucrose, some type of sugar. And water moves toward the area where there's more solutes. So if we look at our first environment here, let me bring up a yellow. You can see this term right here. In this first cell that we have, inside the cell, you can see we've got a couple of solutes. Outside the cell, we have a lot of solutes relative to the inside. Water, okay, these little guys down here, that's water. Water's going to move towards the area that has more solute. So we call this type of environment, we call it a hypertonic environment. So it's going to have more solute than the cell does. Water moves toward the outside. And what happens is the cell shrivels. So this would be like one of the reasons that you shouldn't drink salt water. If you drink salt water, you can become dehydrated. Your cells will lose water because water is going to move towards the salt. So a hypertonic environment has a lot of solute, think like a lot of salt or some type of substance dissolved in it. Think somebody that's hyper, think they've got a lot of energy. Think hypertonic, a lot of solute. Okay. Our next type of environment is going to be a hypotonic environment. Okay. And so in a hypotonic environment, there is less solute in the environment than the cell. So what happens in a hypotonic environment, you can see this is the inside of the cell, this is the outside. There's more solute on the inside than there is on the outside. So water is going to move towards the solute. It's going to move inside the cell. And what happens when water moves inside, the cell is going to swell. So we're going to say cell swells. If this is like a red blood cell in your body, the red blood cell could rupture, it could burst. When you receive IV fluid actually has it has a little bit of salt in it. It's like 0.9% salt. And so this helps to keep your cells from gaining too much water. So it has a little bit of salt in it. It's going to rehydrate you, but it's going to keep your cells from bursting or lysing. But in a hypotonic environment, cells can swell. Plant cells like this type of environment. In a hypotonic environment, plant cells swell, and it gives the cells turgor pressure. Turgor pressure helps the cell to stand upright, and helps the plant to be, it causes the central vacuole inside the cell to expand, to fill up with water, push against the cell wall, and it causes the plant to stand upright. And then our final environment that we have here, you can see we've got an isotonic environment. We've got an equal amount of solutes, both inside and outside the cell. And so we don't have any net movement of water. Water is going to move because it continually moves, but we don't have a net rush of water to one side or the other. So in an isotonic environment, solute concentration is equal. And we have no net movement of water. So no net movement of water. So to summarize what we've talked about today, we reviewed the cell membrane, the plasma membrane, the different components and their roles. We were reminded about the phospholipid and its nature, its polar and nonpolar nature, and why it can be difficult for things that are larger and have charge to pass through. Transport into and out of the cell can either be passive, where it does not require any energy, or it can be active, where it requires energy. In passive transport, we've got three types. We've got diffusion, facilitated diffusion, and osmosis. In diffusion, we are looking at substances moving from an area of high concentration to low concentration, or another way that we say that is moving down the concentration gradient. And the things that can pass through the cell easily are small, nonpolar molecules like oxygen and carbon dioxide. And they're moving from an area of high concentration to low concentration. However, diffusion doesn't have to have a cell to happen. We can see it in things like food coloring in water and spraying something like perfume in the middle of a room. The molecules will continue to move until they reach a state of dynamic equilibrium, which means that they're all spread out, yet they still continue to move simply due to their kinetic energy. In facilitated diffusion, that means that we have diffusion with help. So this is going to be for those things that cannot easily pass through the plasma membrane, things like ions or polar molecules like sugars. They're still moving from an area of high concentration to low concentration. We can still say that they are moving down the concentration gradient. They just have to do so with the help of a protein because they have charge and they are not able to pass through the fatty acid tails. So we would expect the exterior of these proteins to have amino acids that have R groups that are nonpolar in nature that can be next to those fatty acid tails. And we expect the interior of the plasma membrane to have charge. That's going to help the polar molecules and ions to be able to pass through that protein with ease. We have two types of proteins, channel proteins and carrier proteins. Channel proteins are just like a little tunnel that goes through the plasma membrane, while carrier proteins change shape as the molecule goes through. Let's make sure that you actually wrote that down in your notes, because I didn't write it down, I just said it verbally. Let's make sure that we're writing down that channel proteins are going to be like a tunnel that is open where molecules can pass through, and carrier proteins change shape as substances pass through. And then finally, we looked at our third type of passive transport, which is osmosis, which is the diffusion of water. Water is going to always move towards the area where there is more solute. If an environment has a lot of solute in it relative to the cell, we're going to say that it is a hypertonic environment. So water would move out of the cell towards the high solute concentration in the hypertonic environment, and the cell will shrivel. In a hypotonic environment, that means that there's more solute inside the cell than outside. So water is going to move towards the solute, which means the water moves inside the cell, and the cell will swell. It can even rupture or lyse, unless it's a plant cell, which plant cells like it because they're able to fill their large vacuole and push against their cell walls, have turgor pressure, and stand up. And then in an isotonic environment, we have equal concentrations of solute both inside and outside the cell. And so we have a continual movement of water, but no net flow. So this is passive transport, and I hope that this video was helpful to you.